Hi, everyone. Hello, hello. I think we're going to get started so that we can stay on schedule. I think we have, welcome back. And I just want to make sure that people have the number up for the CME at 47767. We have a lineup. This is a lineup for next week. We have a faculty retreat next week. There's an email that went out from Bianca's. If you haven't responded yet, please respond. Then there's a couple of grand rounds interspersed with election day break. And there's a faculty meeting at the end of November. I'm going to hand off the introduction of the speaker to Fadi. And with that,
yeah, so it's a great pleasure to introduce our speaker today, Dr. Colleen Clancy from UC Davis. Dr. Clancy is a professor in the departments of physiology, membrane biology, and pharmacology there, where she also holds many other distinguished titles and, in fact, wears many, many hats. I don't know where you find all that time, but I'm gonna just list a few, or else we'll be here for a long time. Dr. Clancy is Associate Vice Chancellor for Academic Personnel, Director of the Center for Precision Medicine and Data Sciences, the Rolkin Endowed Professor in Health Innovation, and the Interim Chair for the Department of Cell Biology and Human Anatomy. But the title that means most to me is that I get to call her my very close and good friend and confidant for nearly 30 years. Colleen and I met as graduate students in Cleveland at Case Western way back when, back in 1995. So sorry, I'm dating us here. I was in biomedical engineering, she was in physiology, but we really were training in the same center. We joined sister labs, so to speak, that were in the same center. And so I got to really witness firsthand her remarkable innovation, her productivity, her creativity. And just to punctuate that a little bit, while her colleagues, her classmates were still trying to figure out how to turn on and off pieces of equipment, Colleen managed to publish a landmark study in Nature, which really set the standard in her field. After she earned her PhD in 2001, under the guidance of Yoram Rudy, she moved to New York City, to the Department of Pharmacology, to work with Rocky Cass. And together, they really defined the molecular basis of long QT syndrome, as well as studied drug channel interactions using computational tools that she developed. She moved to to Weill Cornell in 2005 as assistant professor, was there for a few years before moving to Davis, where she really not only rose through the ranks to a full professor rapidly, she got to establish really a premier computational cardiology program that's really widely viewed as being second to none in the nation. So Colleen, we're delighted to have you visit with us today, and I look forward to your talk.
Okay. Can everybody hear me okay? Okay. So Fadi, I just want to say thank you so much for such a kind and generous introduction. It's not entirely accurate, for the trainees and the audience. The truth is, when I joined my PhD lab, I really had no skills under my belt to join that lab. And so pretty much on a daily basis, I would go to Fadi like in a complete panic saying, can you go ask Robin for that code that I need so that I can try to figure out like how to do this? Because I was so embarrassed that somebody was going to realize that I had no idea what I was doing. But, over time, I managed to pick it up, and here we are today. So I'm super excited to share this seminar with you today. I put this together pretty recently, and it has some historical elements to it, but mostly it's what we're thinking about right now, what the future could be. And so I hope that you find the message, inspiring and maybe relevant and applicable to your own work. And I had such an amazing day today. Every single person that I talked to, I could think of 100 ways to collaborate with. And so I hope that we will have an opportunity to work together at some point. So I'm going to start with this quote because I feel like it's such an apt description of what we're trying to do now. Every once in a while, a new technology, an old problem, and a big idea turn into innovation. And I love this quote because, wow, we have really arrived at a moment that 2 years ago, I couldn't see coming. I don't think any of us could. The growth and explosion in artificial intelligence and deep learning has brought us already to a place that makes things possible that we couldn't have imagined just so recently. So I want you to think for a minute, what if we started to really address complex biomedical problems by thinking about them from every scale of the system? That's not historically how it works. I've been in a lot of basic science departments over the course of my career, and there's a lot of faculty in those departments, brilliant, contributing members who focus on, one scale of the system. So focus on protein folding, maybe of their favorite protein of interest, or they spend a career patching the ion channel that they love the most, which, I get that. I love ion channels the most too. It's the love of my life, actually, ion channels. Or maybe they're focused on cardiac myocytes and just thinking about how to study those, or maybe they're an epidemiologist and they're really thinking about social determinants of health. But what if we brought all of those scales and all of that expertise together? So an example of the kind of complex problem I'm thinking about is opioid use disorder. I know this is a cardiology group, but bear with me. Not only do we need to think about how to develop new non-opioid medications, we need to think about every scale of the system, cardiac disorders that result from chronic use of opioids. And then what about opioid use disorder as a disease of despair? So how do we really understand what makes some neighborhoods prone to large affect with addiction? And then what is it about some neighborhoods that make it less likely that people will successfully receive treatment or complete treatment? Another example that you could easily think about is heart failure with preserved or reduced ejection fraction. These are extremely complex biomedical problems that can't be solved by just thinking about one scale. So why now? I love this quote too. Physics is to math what AI is to biology. Biology is the largest quantum machine of all. But now suddenly we're on a cusp where we can grasp the inner workings of that quantum machine and really try to disentangle and understand it. All of us are doing what we're doing because we feel this need to engage in alleviation of human suffering, right? That's why we're all here. That's what drives us. But I would challenge you to think about how badly we need new approaches in technology, and that all those mouse models that we've made where we've cured Alzheimer's many times over haven't really been the panacea that we thought they might be 25 years ago. So how now do we move away from reductionism and pivot to leverage and apply the power of AI? And so in my laboratory, but also in the Center for Precision Medicine, I'm really challenging the members to think about how do we really get to that cutting edge and develop new approaches, new technology to solve bigger problems on a bigger scale. I think about this as convergence science, where we're moving away from one cell, one organ, one disease, one organ system, and really think about how can we develop new ways to tackle multiple diseases in the same pipeline. And it's not just us who's thinking about it. This is one of the recent connection puzzles from the New York Times. I'm sure you all do this every night before bed. And look, multi-scale was the yellow category of the day. So well-known phenomenon. One of the things that I really wanted to do was to create a unifying framework for the Center for Precision Medicine, under which we could all operate with that framework in mind. And I was thinking a lot about digital twins over the last couple of years. These are broadly used like in aerospace engineering, where Boeing ships their turbine engine with a digital twin so that the user, the engineers, can optimize the performance of their engine using the digital twin and really try to make it work the way they want it. But they've been relatively underutilized in the biomedical space. And in recognition of this, the National Academy group published a consensus study report just over this last year defining foundational research gaps and future directions on digital twins. And I attended a few of these meetings, and one of the things that I really loved was the levity that the organizers brought to those sessions about the importance of being realistic. Okay, so we're not going to build digital Dr. Akar this year. That's not going to happen. It's way too complicated. I know, exactly. Nor would we want to. No, I'm just kidding. But, we can start to put together the building blocks and the innovation around the intersections of digital twins at different scales of the system. And so what is a digital twin? Again, in the consensus study report, they developed a loose definition of a digital twin that's not constraining, but I think includes a lot of important elements that need to be considered. And so the most important thing is that for the physical counterpart that we want to represent virtually, there's a human in the loop as part of that decision making. Okay, so AI is not going to take us over just yet. There really does need to be a human, ideally with content expertise, that can tell us whether or not the predictions of the digital twin are realistic, and if the data used to build that twin are the right data. Obviously, for all of you that do computational modeling, you know that verification, validation, and uncertainty quantification has kind of become like the terms of the day, and a lot of the grant programs require us to write a section on that. And then the ethical implications also of a digital twin. Who are we leaving out? What are we not considering? What are the things that could negatively impact people if we don't consider ethical implications of our work? So I want to start by kind of talking through some examples of digital twins that we can do right now. So like the current state of biomedical applications of digital twins. So this is a cartoon depiction of the industry drug discovery process. And many of you are probably familiar with this. It starts with lead discovery and then medicinal chemistry, in vitro studies, in vivo studies, and clinical trials. And all of those pieces have to be completed at various points in the FDA approval process. But we can actually, at this juncture, develop a digital twin of that process, where each step in the process can be represented by a digital twin. So lead discovery, just 2 weeks ago, there was a paper in Nature Communications called Rosetta Virtual Screening, which is a super high throughput screening on digital proteins of massive drug libraries. AI-based tool, couldn't have imagined it a year ago, but here it is. The paper is out. And one of the authors on that paper is my associate director in the center. Medicinal chemistry, where we can build proteins in the computer now with tools like AlphaFold that allows us to not only construct proteins from the protein database, but also construct de novo proteins from predictions that are made from the huge library now of known proteins. In vitro studies that can be done in the computer, in silico in vitro studies, I think about those, and in vivo studies as well. Some of you here in the audience are developing whole heart models already, and those could be the basis of an in vivo screen where we think about the geometry of a male heart versus a female heart and how that might impact arrhythmia inducibility, for example. And then finally, clinical trials in a digital twin. I'm working right now with the group in the center on how to develop a model of virtual clinical trials that could become a core part of our CTSC. So not only is each one of these steps, not only is it possible to replicate each one of these steps in a digital twin, but actually the entire process is a digital twin. So you can put these together in any variety of ways and build the digital twin for the prediction that you want. So one of the most powerful tools that has come out in the last 20 years is cryo-EM. You're all familiar with that, where you can actually experimentally measure the protein structures from any variety of proteins. And there's been lots of studies that have come out. You remember the landmark study from Rod McKinnon, who ultimately won the Nobel Prize for resolving the crystal structure of the cardiac potassium channel. Actually, it was the bacterial potassium channel first. And so this is a common technique now, and a lot of institutions have invested in these, which is a pretty major investment. It's not just the original $5 million for the instrument. It's actually the staffing and the preparation of samples that needs to occur to get a good enough data collection to allow for a map reconstruction model building and structure analysis. And one of the limitations of cryo-EM is that you don't actually know what structure you're getting. You're just getting the structure that was, caught in the crystal under the conditions that you inputted. But AlphaFold 2 from Google DeepMind is essentially a digital twin of cryo-EM. All those cryo-EM structures that are now in the protein data bank can be used by AlphaFold, a deep learning mechanism, to start to now predict protein structure. I remember when I was an undergraduate, I was a math major, and I thought, I am going to work on the protein folding problem. That's what I want to do as a graduate student. I had no idea what that meant, and I had no idea how daunting that would be. And it was so daunting until like 2 years ago when AlphaFold 2 suddenly showed us that it probably can do de novo protein prediction. And if you look at the trajectory of protein folding prediction, it pretty much remained flat for 10 years in terms of accuracy of prediction. And then all of a sudden, everything changed. So one of the amazing things about AlphaFold 2 is that it can actually take a protein structure and make predictions about the conformation of that protein in different configurations. So you know that proteins are dynamic molecules. They don't exist in just one conformation. So let's go back to my love of my life, ion channels. We know that they exist in multiple discrete states. And the reason that matters is because drugs bind with different affinity to different states of the channel. And so, for example, a drug that binds with high affinity to the inactivated state of the hERG channel is known to cause more cardiotoxicity than a drug that binds to the open state. But until recently, we really haven't been able to assess those different conformational states using cryo-EM, although people have tried. We're never quite sure which state it's in. But Google AlphaFold 2 now can do that. And Kwa No, who was a PhD student in my lab, recently published a preprint showing that Alpha 2 can plausibly predict the hERG channel conformation in open, closed, and inactivated states. And the cool thing about it is that there's been so many studies on the hERG potassium channel that there is just myriad data out there, mutagenesis data, electrophysiology data, drug data, that allows us to confirm that those predicted structures are likely to be the real structures. I'm simplifying it a little bit here. Actually, it turns out that AlphaFold 2 creates lots of different structural predictions. Those get binned into likelihood scores with various free energy scores. And we actually think that a number of those that are relatively low energy probably represent transitional states between open, closed, and inactivated states. And that's an area that we're actively pursuing now. So what does it mean really to have a digital twin, like of a cardiac myocyte? I just want to be clear what that means. I realize that I'm using my mouse on my screen. Is that, okay, it is the same. Okay, that's good because a few slides in here now to recognize that mistake. So this depiction of a digital twin is really like an asset or an element that has a lot of different pieces inside of it. And so it has data, right, that's used to develop the model, to parameterize it and constrain it. It has source code, it has computational workflows. Sometimes you want to run one piece of the digital twin on a GPU-based machine and another piece of the digital twin on a big CPU-based cluster. And you can actually write that into the workflow to allow it to send different jobs to different resources. And then maybe some deep learning analysis tools that can be used to ultimately make the prediction about what's most important for the morphology of this action potential, let's say. I think one of the really important pieces of developing a digital twin, and this is what we're really working hard on now, is to think about the design elements associated with that. So how do we build something that we think can be almost infinitely expandable, modular, and can talk to other digital twins? So we really spend a lot of time on that design phase. It's really kind of an engineering problem. Okay, but it's not just about building a twin at each scale of the system. It's about thinking of new ways to connect the scales. And I think of all the things that we're doing and others are doing, maybe one of the most interesting pieces is thinking about how do we connect, for example, the atomistic scale simulation, which is happening at the spatial scale of angstroms and the time scale of nanoseconds and picoseconds, maximally maybe a microsecond if you're running it on the largest national computational resources like Anton. How do we take something like this and connect it to cellular or tissue level behavior that's occurring at the scale of centimeters and seconds? So this is a problem that we've been working on, and I'm not exaggerating, for more than 10 years. It started with a conversation in my office with Vladimir Yarovoy, where he was working on structural prediction and we were working on functional prediction. And I was like, there's got to be a way to marry these. And we kind of threw some ideas around, on the whiteboard. And finally we said, okay, there's really no way to connect these in terms of time. Like it's just way too many iterations at the atomistic scale to even get to like a millisecond. We're just never going to be able to do that. But what if we could use atomistic scale simulations to make predictions that could help us to define parameters for the higher order models? So let me tell you what I mean by that. First thing I want to do is just walk through each of these data points on this table. I'm just kidding, totally not going to do that. Now, so just kind of the overview here is that this is showing a hERG potassium channel in the open state. And what we're doing here is a molecular dynamics simulation with two drugs, dofetilide, which is a known hERG blocker and has pro-arrhythmia potential, and moxifloxacin, which is often used as a positive hERG blocker control in experiments, but has no major risk factors, so it's widely used. So why is that, right? Why would they both be hERG blockers, but only one of them seems to have proarrhythmic potential? Can we use kind of atomistic scale approaches to discern the mechanisms of binding that will allow us to say why one is toxic and one is not? And just for the chemists in the group, we didn't just consider one species, we considered neutral and charged dofetilide and neutral and charged and zwitterionic forms of moxifloxacin. I learned the word zwitterionic while we were doing this project. So what we were able to do is to put the drug inside the binding site. We know where the binding site is in the pore of the channel. And I'm not going to show you like all the simulations that we did to confirm that. But pretty much if you flood this protein in a bilayer membrane with drug, you see no other access of the drug. It doesn't go through the membrane, which is not true actually for sodium channels where we see drug moving through the fenestrations into the pore of the channel. Here, the access points seem to be into the pore. And then what you can do, and this is a roughly representative, is to divide up that entire pore into windows and force the drug to sample in those windows, and then calculate the free energy of binding all along that trajectory of the pore. The more left this peak is, the stronger the binding, the lower free energy of binding. And it turns out that the free energy of binding is proportional to the KD of the drug. And then what we could do is run diffusion experiments in the computer to ask how long does it take for the drug to get into the binding site just through the regular access pathway. Typically, it's the drug coming off that's rate limiting. So the drug going on is, it looks pretty similar regardless of the species. But we can consider that as K on, and we have KD, and so we can calculate K off. Once we have K on and K off, we can actually introduce those parameters into function scale models of the gating ion channel. So that was our way to find a connection between something we could generate from the atomistic scale simulations and something we could use in millisecond and second time scales. And then once we do that, we can take that drug channel interaction and put it into the computational model of the cellular action potential. We can couple those cellular models together end to end through simulated resistances to represent gap junctions. And then we can compute the signal average in time and space of electrical activity across that tissue to generate a pseudoelectrogram that we can compare to the clinical data that many of you produce. And so Pei-Chi Yang, who I've been working with for 20 years, took those computed parameters from the atomistic scale simulations and put them into our model of dofetilide and moxifloxacin. With dofetilide, there was one free parameter left that we couldn't constrain. And so we did fit that to the experimental data. But for moxifloxacin, as you can see, there's no closed loops in those models. This is for you guys, the model aficionados here, little microscopic reversibility lesson. Because there's no constraint on that, we could actually use those parameters directly from the atomistic scale model and look at the fit to the IC50 data. I got to tell you, I didn't believe it. I was like, there is no way that fit like that. But we've actually done it multiple times since with other drugs as well. And we're kind of shocked that it does really seem to work. But that's not what tells us it works, okay, that we can reproduce the experimental data. What tells us it works is when we can use the clinical data to validate the output of the model. So this is really the gold standard. What I'm showing here is the model predicted concentration dependence of the corrected QT interval from simulations and then some experimental data points because they only use that one concentration. And then in a variety of patients, the change in corrected QT interval following application of dofetilide. And this is from 2 different clinical studies are the lines and the dots are the simulations. And then the relative change in the slope of the QT interval compared to the preceding RR interval compared to clinical data. And then finally, a whole bunch of studies with moxifloxacin from the clinical data And our simulations, you can see, fall on top of those. And then finally, as part of this study, because some reviewer requested it, even though it felt like we'd spent $10 million on that study already and we'd really done everything that we should do, they still wanted some machine learning in it, even though it was like 2019 and it wasn't even like really a thing there, but whatever, we did it. So Pariaga Safari, who was in my lab at the time, a really talented postdoc, was able to show using a variety of machine learning methods what was most important in predicting arrhythmia likelihood following application of dofetilide. But beat-to-beat instability, interestingly, was the parameter that emerged from that machine learning classification. Okay. So what else could be used? I tried to pick some examples of things that would be relevant to the engineering group and the cardiology group. You know, another thing that can be done right now is programming neural stimulators through digital twins. And so we were very lucky to receive some funding from the Common Fund a few years ago through the Stimulating Peripheral Activity to Relieve Conditions. I talked to some of you today about sympathetic nervous system stimulation. And one of the things that we did was to build a layered model of the autonomic nervous system based on a variety of data, both from our collaborators, but also from the experimental literature. And we were able to validate some of the behaviors of the various firing dynamics that emerged through that layered network model. And this was really through a collaboration with my longtime collaborator, Tim Lewis, in the mouth department. And again, surprising sort of emergence of the dynamics of tonic accommodating and phasic models from the network model. Because we were all into like connecting the scales, you know, the atomistic scale to the cellular and tissue level scale, Igor Vorobyov actually carried out some simulations in the beta adrenergic receptor to get at the rates of norepinephrine binding to the adrenergic receptor. And we were then able to utilize those rates in a functional model of the beta adrenergic activation that was added into both a sinoatrial node model and a ventricular tissue level model, and calculate the electrocardiogram for that and show how we could apply parasympathetic nerve stimulation and sympathetic nerve stimulation. And I won't go into all the details, but in the paper, there's a lot of different variations on that. And we validated some of that with experimental data from Crystal Ripplinger's lab. But the most maybe important result here was what we could show is that in the presence of really strong sympathetic surge, we could readily induce an arrhythmia in this human simulated heart. But if we were to add parasympathetic nerve stimulation and then withdraw that stimulation in the presence of high adrenergic tone, we could cause elimination of those arrhythmia triggers, even in the diseased heart. And this was a representation of heart failure. Just want to say, I know I'm talking fast and I'm going through all these studies. This is like 15 years, okay? So it didn't just happen like one day to the next. But you can imagine how now this model could easily be transformed into a digital twin where we think about comorbidities that a patient may have and or other attributes like drugs they might be taking, ACE inhibitors, beta blockers, flecainide for their atrial fibrillation, and then use all of those parameters to make a prediction about how that device should perhaps be tuned for that patient to reduce or prevent or terminate arrhythmias. And it's kind of a big deal because, you know, they implant like neural stimulators for everything now. And part of the reason for the SPARK program was like, if you get a neural stimulator implanted for your gut motility, like nobody really knows how it works, just so you know. So the SPARK program, they really wanted people to understand the mechanisms of the efficacy of those neural stimulators. It's funny, in the last year, I've been at the doctor and 2 different doctors have offered me a neural stimulator for problems that I didn't think were really that big of a deal, but they're definitely out there and they go through a much more lax FDA approval process. Another current application of digital twins is using the iPSC-derived cardiac myocytes that are being used experimentally now for drug screening. But what we were able to do in the laboratory was to show how we could develop a gigantic synthetic data set, use that to train a machine learning model that could parameterize every single one of these components in a cardiac cell model. We haven't published that yet, but we simplified all those model components into really simple Hodgkin-Huxley forms and then built a training set with simulated data. The benefit of doing that is you know what the input is. So you know if the model is working because it should get you back to the original parameters that you put in. I think there's a lot of potential utilization for that kind of approach in the spaces that we live in. And once we build the tool, then we can apply it to real iPSC cells and extract the required information. It's actually a single voltage protocol, which was actually part of a nested loop of the machine learning algorithm. So it learned what the protocol needed to be able to get the parameters out of the model. And then we can do it on a real cell and create digital twins of all of your cells. That includes all your natural variations. And then you might say, well, who cares? You know, they're just simulated iPSC-derived cardiac myocytes. But we also built a machine learning algorithm that can translate between those immature iPSC cells and the adult cardiac myocyte, and tested that even in the presence of drugs and showed that we could validate it with clinical data. Finally, the most recent study that I would say is current state is building on a recent study showing that inhalation of flecainide can be used to acutely terminate atrial fibrillation, which is kind of like a big deal, you know, because for all of us that live here in New Haven or in Sacramento, you know, we can have an arrhythmia episode and we just go to the emergency room and they cardiovert us and then we follow up with the cardiologist the next day. But in terms of like global health equity, the vast majority of people don't have that option. And if there were a way to chemically defibrillate, like on the spot, it could really potentially improve a lot of lives. What we were able to show using a digital twin is that actually we know that flecainide can come with risks, including really negative inotropic effects. It can actually cause almost a stunting of the heart. And what we showed is that we can actually lower the dose of flecainide quite a bit by adding a tiny bit of butylide on top, not enough to cause any safety pharmacology risk, but enough to enhance the effect of flecainide. Okay, so that's all well and good. That's kind of the current state. Where are we? But here's the stuff that I'm actually super excited about, and I hope will stimulate a discussion. What we really want to do now is think about convergence. How do we move beyond thinking about, you know, atrial fibrillation? What if we could find common elements among pain, atrial fibrillation, and breast cancer metastasis? Well, our preliminary data suggests using large language models, there are actually tons of shared biomarkers across those diseases. And you're never going to believe this, but one of them is the voltage-gated sodium channel, the love of my life. So calcium channels are also a number of proteins in the oxidative stress pathway, and a number of other biomarkers that came up in our original analysis. So what that means is if we have a common target, we can develop a pipeline to try to identify therapy across all of those diseases at the same time. So how are we going to do that? Well, as I mentioned, you know, we started with a large language model, but then what we started to do was use a natural language processing approach to mine all of the PubMed text to validate what came out of the large language model, and also to find other important attributes in the pathways that will be really important to build functional scale models to understand the networks. And then to use a computational genomics approach to try to identify common variants across different ethnic groups that may be relevant both to disease severity and or phenotype or to drug intervention. Once we can identify coding and non-coding variants, especially with coding variants, we can actually take those mutations or polymorphisms or variants of unknown significance and introduce them into the structural models that we can build very quickly in AlphaFold 2 and ask, are those changes in places relevant to protein gating, or are they in the pathway of a drug binding site? As I mentioned, one of the things that just happened in the last couple of weeks is once we have those proteins that contain different genotypes, we can start to screen those in this accelerated virtual screening pathway. And Vladimir Yarovoy, who has been my longtime friend and partner and now associate director in the center, has really been at the forefront of bringing this to life. Another very important collaborator for us is David Lim, who recently moved to UC Davis from UCLA. And he has been working on trying to understand social determinants of complex diseases like heart failure with preserved ejection fraction. And so David has joined us now. We're meeting with him every week so that we can bring that neighborhood scale into the digital twin pipeline. And then what might an in silico clinical trial look like? I mean, it's almost limitless. We have to start kind of with the data that we have, but you can imagine lab tests that indicate, metabolism, that tell us something about the other drugs that a patient is on, that tells us maybe some other comorbidities, connecting that with the genotype, looking maybe at images to figure out, there's some really great machine learning images approaches now. Adam Yala at Berkeley, who has just developed a deep learning approach that seems to do extremely well predicting breast cancer before there's any evidence of tumor in the image. So, that immediately leads me to say, what is the algorithm finding? And I spent quite a bit of time trying to probe that question with him. And he was like, whatever, who cares? It's predictive. And it's like, wow, I really care. I want to know what is the information that it's using to make that determination that really has a high level of accuracy, much better than anything that's being used in radiology right now. What about liver function and kidney function? Can we incorporate those kinds of things into a virtual clinical trial as a evidence or as a proxy for drug metabolism? And then other organ systems that could be relevant to a disease of interest. And finally, that digital twin neighborhood. And so with that, I'm going to end. I hope that we can have some spirited discussion. I never have worked alone my entire life, and I can't tell you how much this talented group of people has brought to the research program that we're all developing together, but also has brought to my life personally. These are really, really wonderful, amazing people that shock me every single day. And finally, just to kind of acknowledge over the years, the funding sources that have kept us alive. Thank you.
Thank you, Colleen. That was a tour de force discussion in computational cardiology and medicine, and as it relates to past and future and directions. So do we have any questions from the audience to start? John.
Thank you very much. Aging is the major risk factor for aging. And aging certainly affects all the way. And so if you age and then use that to predict mortality, morbidity, and then potentially mortality.
You don't even know how much I love this question. So, one of the things that one of my students is working on right now, I'll just point her out, Hannah Zakowski, who came actually from Connecticut. She did her undergraduate in mechanical engineering at Trinity and now is a BME student at UC Davis. She's working on developing a model of oxidative stress in the cardiac myocytes. So trying to integrate oxidative phosphorylation pathways and their stress by ROS, connecting that to contractile elements in the cell and also electrophysiology. So that would sort of be at the cellular level digital twin. But, if you think about the protein level as well, we've thought a lot about this. Like, what are the modifications that we might expect to see in the lipid environment and or in the protein environment? And so I actually think if we had a comprehensive understanding of data and the data itself, we could absolutely incorporate that. What Hannah is working on is cardiotoxicity of radiation exposure for cancer treatment. But the model is the same for tyrosine kinase inhibitors, and it's the same for aging, actually.
Fascinating talk. So you showed this AlphaFold and its use for actually showing the structural change. But a lot of these mutations that actually they have in common are not actually in the protein. They are not full. Many of them actually done now. They had a Nobel Prize for the RNA. So what is then the next step?
Yeah, so I think definitely there's kind of two sides to this, right? In the coding region, one of the first things that we're doing is sorting the mutations, identify where in the protein we think they're going to be. And to be honest, we expunge the ones that we don't think have any effect on function or are in the drug binding site. They may be important, but we can't do everything all the time, right? So we have to start someplace. So we're trying to first identify the ones that are most relevant to the structure and what we think would affect gating and or drug binding. For the non-coding, what Hajar is doing now in the center is trying to find a connection between those non-coding variants, and she's using all of us data to understand disease severity associated with those non-coding gene variants. If we could understand that, then we could incorporate some of that into the functional scale models, but we haven't quite figured out how to do that, but we're thinking about it, and I'd love to hear your thoughts on that.
So that's really awesome work, and this is not to say I'm a non-believer. I'm very much a believer, and I think AI is going to get us digital computational analysis will get us to places obviously that we've never been. But there's still an issue of complexity of individual cells in a cell specific fashion, and then obviously the other extreme is the complexity of a human, of an individual, where until we get to a genetic scorecard,
yeah,
which may be epigenetic scorecard as well, but everything that involves the difference between one individual and another, unless you do the clinical trial and see who works and who doesn't, you don't really know. Although even the clinical trials might not get you to understanding why it works in so many ways. But for the AlphaFold 2, that's a great example. It could predict protein structure, but if you don't look in a specific cell type, maybe the cytoplasmic domain is influenced by a protein that is cell type specific, that then affects the channel, or integrins are a great example.
Definitely.
Cytoplasmic interactions influence how the structure, the external structure is. I mean, those are like, how do you do that? How do you get to all that?
Yeah, great question. So we're not going to get to it in our lifetimes, right?
Yeah, that's
But you raised such.
I assume that anyway. Easier for me to say that.
I don't know about that. I'm really tired. No, you raised such an important point, and I think I always talk about the most important thing we can do is to define the context of use of the model that we're building and the purpose of the model that we're building. And is the model fit for purpose that we've predefined? So for example, what I showed you today is we're interested in how maybe variants, genetic variants, modify drug binding. Well, you know what? We deliberately selected a target where we know where the binding site is. It's not just one binding site. There are actually toxin binding sites all over the channel, but they've been well mapped. So we're using those known things as deliberate examples that we've biasly selected to demonstrate a proof of principle of application of the technology. Am I interested in screening every drug on the planet on every protein target? I am not. I'm interested in publishing that first way to do it and then moving on to a new problem. So, but I think, in terms of utilization, others may well be interested in screening on everything. Now, what you mentioned is also really important. Turns out AlphaFold 3 allows you to actually predict the structure of proteins in complex. This is one of the things I've always wanted to do, is to think about, so let me get like a little philosophical here for a second. There's so many processes that we know happen in the cell: phosphorylation, dephosphorylation. Let's just take those two examples. But if you look, for example, at the concentration of phosphatases in a cell, and we've done stuff where we set up like a stochastic simulation and we look at the probability of a phosphatase running into a target, the probability is 0. So that's like the punchline that took a lot of simulation to get to that. There's something much more concerted happening. And I think that concerted thing that is happening is proteins are actually bundled together in spatial proximity so that reactions can occur when and how they need to occur. What I would love to be able to do in my lifetime is to model some of those and then be able to use tools like AlphaFold 3 to try to predict the rates of change in that type of process. So like phosphorylation and dephosphorylation, depending on the proximity of those two things together. We don't have any way really to do that right now. But again, I think it's all about context of use. Even with the cardiac myocyte, does it contain everything? It doesn't. But does it contain what we need to be able to predict the effect of the drug on the electrophysiology in the ventricular cell wall going from endocardium to epicardium? It appears so because we can validate it with clinical data. There sure are a whole lot of things it can't do right now. But these guys are going to help me.
Let me follow up a little bit because I'm not necessarily sure I agree with that last statement. There's a lot of other cell types that will play a role in modulating conduction that are being maybe missed. So my question is, we're at a stage where you can make very bold, powerful predictions, and that might be based on incomplete data sets, maybe based on data sets that are available today, missing critical insights that we don't have experimental ways to determine. And when do you become concerned that some of the predictions that are made in a certain context, say a normal myocyte, would not hold muster when the myocyte is remodeled or
So everything you said is like the stuff I lay awake at night worrying about, right? But again, it's about defining context of use. So I would never presume to say that predictions of a drug in a normal cardiac myocyte that's built from normal data would relate in any way to a remodeled myocyte. But let me just say, one of the most important things, I think, is domain expertise. I can quickly look at a data set for an ion channel, for example, electrophysiology data set, and say, that doesn't look right. I'm not going to use that data. I know that an activation curve does not have a V-half that makes sense to me. Now, if I didn't know that, then I would just use that data to build a model. And we see a lot of that. For the last 5 years, I sat on the JACC clinical electrophysiology. I was associate editor there. And we would just get dozens of papers where people take data and push them through machine learning algorithms. And they say, okay, well, this causes this. But without any real familiarity with the data that you're using for the training set, you actually have no idea if you're identifying a cause and effect or not. So I can't stress that enough. All these tools are useless if you don't have any like internal quality control, which is really domain expertise that somebody can say, this data looks right or the output looks right or wrong. Does it make sense? In some way to validate that.
One follow-up question, sort of along the same lines. This is really dependent on open science, free sharing data. The libraries use refinement for this or good representation, but it's at odds with philosophically a lot of our business models from industry and academic thinking. So how, again, there's a solution to this, but what do you posit will be necessary going forward to achieve what the future?
Ain't that the truth? Everything you just said. So I have a theory what's going to happen. Nobody's going to share all the stuff voluntarily. Here's what I hope is going to happen. I think NIH is going to increasingly require open sharing of data in standard forms. But clinical data is also a huge I can't even get access to our own EMR at UC Davis. I run precision medicine, you know what I mean? But there's so many concerns about privacy. But I really believe the payers are going to start to require that. They're going to want to do their own analysis on our, pooled insurance groups and how well we're taking care of those patients. I actually think that would be a really positive development if the payers pressured the medical systems to make those data open and public and de-identified, of course. But there's got to be external pressure to do that because there's no other way to do it. And I don't know. I would say we all have enough money, don't we? I got enough money. I mean, it's like, what else would I want? Right? Like, I think we all just like need to find like the value proposition in what we're doing and say, like, I'm going to make these tools available to all of you because I want you to do the next better thing with those things. But, I'm like an optimist and,
I don't think it's going to happen with that regulation. I think that's
I think so too. But that regulation it has to come either from a government entity or from the other government entity, which is private insurance in the US. The only other entity with as much power.
Any other questions? Oh, there we go. Shabana.
Most of us at the end of the day are so how should this AI system prioritize expanding its insight into how the mouse technology is working versus just sticking ahead and focusing on the
I love your question. It's one I've thought about a lot. I think even with a mouse model, context of use is extremely important. Are you going to study repolarization abnormalities in your mouse? Probably not, right? Are you really, how many more dogs are we going to paste to death? Kind of have an issue with that because I really love dogs. But Fatty, I'll tell you about the time we were going to get a heart from the dog and I cried and I was like, no, we can't do it. And then we didn't do the experiment. And then I'm pretty sure he went back later and did it. So it was a really cute golden retriever. But really, that atrial model of pacing, that's another thing that keeps me up at night. Is that really a good model for atrial fibrillation? I mean, I don't know. We all have to be thinking a little bit more carefully about what we're using the models for. And is the model appropriate for the thing that we're doing? Maybe there's lots of stuff like, I don't know, dermatitis or whatever that the mouse model is perfectly good for. I don't know. But I think we really do need to think that through carefully. And, in the UK, they have really high standards for animal use. And you have to justify up and down why you're going to use an animal to do the thing that you're going to do. But you can always come work with me and then you can just do the digital stuff. See? Problem solved.
Well, I think let's thank the speaker for a delightful talk. Thank you very much.
And I opened all the waters. I don't know why. My brain is out.